Hello, so the next video now, in the previous video we talked about the level of biodiversity. I defined the biodiversity and I told you the three levels of biodiversity. That was the species diversity, genetic diversity, and then we talked about the ecological diversity. Then I told you about the species diversity in detail. We talked about the species richness, the abundance, species richness, and species evenness in the species density. And then we talked about the genetic diversity, the number of genes and the number of alleles per gene discussed. And then I told you about the ecological diversity in the sense of complex food web, more way of interacting with the environment. So there are more food chains, more complex food chain, food web, more biodiversity will be there. Now we will discuss how can we assess biodiversity, how can we estimate the index of biodiversity. I mean, if there is a huge area, a lot of area, and it's like a forest of so many kilometers square, and you want to calculate the abundance or richness of some species in that forest, so it's a very difficult task. It's not easy to calculate the biodiversity of the entire population, of the entire communities. So what should we do then? If you're not able to calculate the biodiversity, I mean, how many species are there, how many species are there, and how many individuals in each species are there, and how many ways of interacting are there. It's not an easy task for the entire population, for the entire community. It's a very difficult task. So how can then you calculate? So the common procedure, normally we have to take some samples, because on the basis of sampling we can conclude, we can estimate that yes, this population may consist of these number of species, and yes, this is about the distribution of that species. Like I'm giving you an example. If somebody asks you to make a survey and move to the different area of Karachi, I mean like North Nazabad, Gulshan Iqbal, Defense, and blah blah blah. So many districts we have in Karachi, so many area we have in Karachi, and a person asks you to ask, and a person have given you a task to ask the people which type of car they would like to purchase. If a person have like a selling person car, so it's not easy to interact with individual person in all these places in Karachi. It's not easy because it's like more than 10 million, I mean 20 million people to interact with these people in Karachi to ask them. It's not easy to ask. So what should we do? You have to make sampling. You have to make an interview with 200 people in North Nazabad, 200 people in Gulshan, 200 people in Malir, and so in other area. And then on the basis of these people, on their choices, on their interview, you can conclude that might be this entire area like the same car. So that's why we should take into account sampling when you calculate the biodiversity of a huge area, because it's not possible to get information about each and every organism in the entire population, in the entire community. So sampling is one of the technique which we have to do it, and we have to calculate. So our topic should be sampling. So why do we need sampling? Because we are not able to get the the abundance and species richness in a very huge population and a huge large area habitat. Then you have to take some samples to conclude the index. So sampling is a procedure. Now when you are going to make sampling, there should be two types of sampling. Let's define sampling. So sampling is the collection of individuals from a large population, which may be the representative of the entire population. So collection of specimen from a huge community or large habitat, yeah, or a big size habitat as a representative of the entire population. Or you can see the more specific word of the entire community or habitat. So, when we use the total population, and what is the sampled population? So sample is the representative of the entire population. We can say like in this area, it's a very big area here, and we are going to make some sampling of diversity. It's not able to roving throughout the area, but it's so many kilometers square of that area. So what should we do? We do some sampling like from here, from here, from here, from here, from here, from here. So like these are the samples. We have collected some specimen from here, and then we calculate and we divide the total area, and we can conclude it that yes, if this particular number of organisms are present in that area, so we just multiply with the entire area, and we can conclude that this might be the biodiversity. So in more detail, let's move. Now, there are two types of sampling when you are going to do. One is called random sampling. And one is called systematic sampling. Now, what do we mean by these two? Random sampling and systematic sampling. When there is an even distribution in a habitat, the species are randomly distributed, and there is no advantageous or no some selection pressure on certain organisms. I mean, the environment is stable for all the species in that particular habitat. Then we do random sampling, because if there is no variation among the organisms in the entire area and they are evenly distributed, so it's up to you. We can take the sample from anywhere, from here, from there, from here, because these are uniformly distributed, doesn't make any problem. But if they are not evenly distributed and some factor is responsible, and according to this factor some of the organisms are like at one place which are not on the other one, then you cannot do random sampling. I'll give you one example. Like you have a square area, just only 1 meter square, a short area, and there's some insect, and you want to collect these insects, but these insects should be initially anesthetized, so they cannot move from one place to the other one. And this small area, like here, you just want to calculate the number of insects. Like once you make one box here, other box, these are some small boxes from different trays of this short area, and just count the number of insects in each box. You just count the number of insects, and you have calculated like one square box, this one. You say that the insect might be uniformly distributed, so it's up to you. From anywhere you can take the sample. But let's suppose if there's some food stuff here, so obviously more insect will be attracted toward the food stuff, and there's more chances, more probability that a lot of the insects should be crowded here. Now, if you took the sample from here, there will be the very least number of insects, because they are not here. Most of them are migrated, they are attracted toward the food specimen or the food particles. So in this case, a pressure is applied on the insect. I mean, environmental factors, and that's food. So in this case, you can't do the process of random sampling. It's bias now. It's biasness because you are taking a sample from that area where there's no insect. But most of the insects are here, so this is not the true representation of your original population. So in such cases, you have to do systematic sampling. Similarly, if we have a field, and in this field, we just want to calculate daisy plant, like daisy, we just want to calculate how many daisy plants we have, And for this purpose, let's suppose here is a canal where water is flowing from this bank. So that area of the field might be more, that area, a lot of water in that area, it will always be a lot of water as compared to the rest of the part, because there should be less water as it's near to the canal. So maybe if the daisy plant like water, there should be, this area should be rich by daisy because it's near to water. So if you are making samples from here, from there, from here, and you're not taking sample from there, it is also biased because these factors, water is affecting the population of daisy plants at this, at this site. So you must have to take in account either there's some external factors which are affecting the distribution of organism in a habitat, then you have to do the systematic sampling. So this is the difference. When should we have to take an account about the random sampling, and then we have to take about the systematic sampling. So if there is no external pressure and all the environmental factors are in such a way that all the organisms are uniformly distributed, there's no favoritism, or there's no negativism, there's no favoritism and not removal of the organisms, then random, okay, you can do this from any part of the population. But if there are some factors which favoring some population, and you are choosing these organisms which have some favoritism because of the external factors, then you have to do systematic sampling. So we will discuss one by one. Now let's talk about the random sampling first of all. How to assess the random sampling? Now assessment of random sampling. Now there are two methods to do this. There are two methods for calculating random sampling. One is called Quadrant Method. And the other one is called percentage cover. What is quadrate? Let's talk about the quadrate first. What do we mean by quadrate? A quadrate is a rectangle box, maybe made up of a wooden piece, might be a nylon rope, or maybe some cotton, some copper wire and so on. But it is a rectangle box with, not rectangle, I'm so sorry, it's like a square because it may be equal dimension here. Like, let's suppose if this is 1 meter, this is also 1 meter, this is also 1 meter. So it's like 1 meter square is the total area of this quadrate. And when we make some boxes again here, after interval of 10 centimeter, so 10 means we can make 100 boxes like in this way. It's not like 100, I'm just giving you an example, like here and here. So we can construct 100 boxes, each one after 10 centimeters. So one box should be 10 centimeter square. It's like a quadrate. This is called a quadrate. What is the function of this quadrate? We just put this quadrate randomly in a grassland. It is a short area because this is a small quadrate of only 1 meter square. Like your college lawn and your garden at home. Some short area with small species like grassy plant that can be captured, that can be accommodated in these quadrate. So if we place quadrate at one, like we have placed quadrate, let's suppose now here we just put in the quadrate in this. This is your garden area. This is your garden area at home. This is your garden area. and you are just putting the quadrant. The first quadrant I have like here, then I make the second quadrant here, the third quadrant I make here, the fourth quadrant here, and the fifth quadrant here. I total make fifth quadrant means 5 meter square area you have chosen. Because one quadrant is 1 meter square, and there are 5 quadrants, so 5 meter square area you have selected, and the entire area is quite greater. It might be like you can see, like it might be 50 meter square area, big area, but you have collected only 5 meter square area sampling, and that is the now you have to point out which species can exist in this lawn. Your task, somebody asks you, just search out daisy plant and dandelion. These two are species, dandelion species and daisy species. These two different plant species. And we just want to calculate how many daisy species are distributed in our college lawn and how many dandelion species are distributed in our college lawn. We just focus only 2 species right now in the quadrant. Now we see the quadrant like in the first quadrant. This is quadrant 1, this is quadrant 2, this is quadrant 3, this is quadrant 4, and this is quadrant 5. We have placed the quadrant 5 times at different places, and we just count the number of these daisy plants and dandelion species, and just making the result here, like here in the form of a table. So here is the species, and here the quadrates, here the quadrates. Here is the species, here is the species, which may be daisy and dandelion. So in quadrant 1, quadrant 2, quadrant 3, quadrant 4, quadrant 5, we have 5 quadrant here. In quadrant 1 we got only 2 daisy and no dandelion. In quadrant 2 we got 1 daisy but 5 dandelion. In quadrant 3 we got again 2 daisy and 2 dandelion. In quadrant 4 we got 2 again and 2 again here dandelion, and then we got in the last quadrant there's no daisy but we got 1 dandelion. Now how many quadrants we put here? 5 quadrants, and we got daisy and dandelion. Now in this quadrant method we have to calculate 2 important things, two important features in quadrant method. What are these 2 features? One is called species frequency. Species frequency, and the other one is called species density. Species frequency and species density for each one. What is species frequency and what is species density? Basically, species frequency is used, species frequency I'm talking about, how many quadrant possess that particular species, irrespective from their number. Like here, if you talk about the daisy, so daisy plant is present in 4 quadrants. We are not counting the numbers, either 2, 1, thousands, 5,000, 1 lakh, or only 1. So only 1 and 1,000, they have the same weightage, present or not present. So yes, we have 4 quadrants which have daisy plant. Here we also have 4. So if you want to calculate the frequency, now keep the formula here. So frequency of daisy plant, let's suppose the daisy plant, is equal to number of quadrate, quadrates contain the species, the daisy, upon total number of quadrate. And it should also be taken into percentages, so you just have to multiply it with 100 as well. This will be 100. So if you go to calculate number of quadrates, of course 4, total number of quadrates 5 into 100. So it should be 80%. 80% is the frequency of daisy plant. And of course the same should be for the dandelion, because it should also be 4. So we have the dandelion also have 80% frequency. That's for the dandelion as well. Both have the same frequency. Now what does it mean? इसका मतलब क्या है? What does it mean? You know, frequency basically tells us about the distribution of species. It doesn't tell us about the abundance, about their number, about their about their total number. We just have how much they are distributed. Like 80% means these daisy plant and these the dandelion plants are distributed 80% of the area. So they're not counting how much maybe they are just 1, 2, scarcely only 1, 2, 2, 3, 3, something less number, but they cover many area. The distribution, their frequency shows the distribution of a species. How much it is distributed, widely distributed or less widely distributed. So 80% means more widely distributed. It's occupy a lot of the area irrespective from their number. Maybe they're in less number, but they covered a lot of area. They are distributed thoroughly. And now if you want to calculate the species density, is something different. Now for species density, what should you do? You have to count the number of individual of each species in each quadrate. Like if we start from daisy, there are 2 daisy in quadrate 1, 1 daisy in quadrate 2, so means 3, 2 daisy in quadrate 3 means 5, 2 daisy in quadrate 4 means 7. So we have 7 individuals of daisy in 5 quadrants. And similarly here we have 5 to 7 to 9, and we have 10 dandelion in quadrate. So we just have, if you want to calculate the density, for this purpose you have to calculate the value, but I think we have to do it into the next video because this is short of time. We'll see you in the next class.